The title of my talk is Protecting Our Most Vital Assets, Our Genome and Environment, the 50-year legacy of EMS. So what I'm going to do is give you a very brief history of some of the antecedents that happened before the occurrence of the formation of our society, some of the events leading right up to the formation of our society, and some of the early events that happened right after our society was formed. So particularly for some of you that are young in the audience, you might not know any of this history. I would like to see the remarkable history that you are coming into with this society. So here's an outline of the talk. We're going to talk first about the first three mutagens, which all of you know about, but you might not know some of the details I'm going to tell you. We're going to talk a little bit about Oak Ridge, because this was critical to the formation of our society. We're going to talk about the environment and germ-cell mutation, the environmental movement, and some of us in this room lived through the 60s and 70s and can tell you all about that. We're going to talk about the takeoff of the society in 1969, some of the early successes, and the legacy that the society has left to us. Now, all of you know about the first mutagen. Obviously, X-rays reported in 1927 in Science by Hermann Muller while he was at the University of Texas at Austin. What you may not know is that in that paper he has no data, no tables, no figures, no numbers, nothing. He simply claimed that X-rays was a germ-cell mutagen. He actually didn't publish the data until a year later in 1928 in PNAS. And in that same year, Lou Stadler down at the University of Missouri in Columbia also showed that X-rays, and he reported the data, was a germ cell mutagen in corn and in barley. So in 1 year, 1928, we finally had the first mutagen. It was a germ cell mutagen both in insects and in plants. The second mutagen came along very shortly thereafter, and you're going to find an interesting theme here. All of this was happening in Texas and Missouri. Edward Altenburg at Rice University in Houston in 1933 reported for the first time that UV light was also a germ cell mutagen, again in Drosophila. And a few years later, Lou Stadler, again at the University of Missouri, reported the same for maize. So within just a few years, we now had two mutagens. Both of them were germ cell mutagens, both in insects and in plants. Well, finally, the main character in the story I'm going to tell you about this morning, Alex Hollaender, comes into the scene. So at the same time, in the mid-1930s, Alex had the first hint of what today we would call DNA repair. You're going to see a constant theme in my talk here, flipping between mutagenesis and DNA repair, the role of X-rays, and the role of UV. This is critical to the formation of our society and to our science. So Alex had evidence early on in the 1930s that after he UV irradiated fungal spores, if he held them for a while before he plated them, and then plated them, they survived longer. So he pointed out the suggestion that the possibility of recovery of irradiated bacteria is not entirely excluded. He didn't know what the nature of that repair was, but he knew that there was some repair of something going on to improve viability. His real accomplishment came just a few years later in 1938, where, again using UV, he suggested that nucleic acid is the genetic material. It's UV, and he did this one at NIH. The previous work was done at Wisconsin. He showed that UV induced the maximal mutagenesis in fungal spores at 265 nanometers, and the maximum absorption spectrum of nucleic acid is that, suggesting to him that UV acted on nucleic acid to produce mutations. So he proposed the notion that the mutagenesis that UV was causing was due to UV's action on nucleic acid, and that nucleic acid must be the genetic material. Now, I have a surprise for you. There aren't but 10 people in this room who knew Alex Hollaender and talked with him. I'm one of them. The rest of you never met him, you never heard him, and you, other than seeing his picture, you know not a whole lot about him. But I'm going to show you him right now. I put together a clip of an interview he did in 1983, right here in Washington, D.C., down the street at the Cosmo Club, where in fact he discussed this really important discovery that he made in 1938, and I hope you enjoy it.
And when I counted these mutations, the maximum was always around 2650, that's the resonance line of Mercury. This is the wavelength which is most highly absorbed by nucleic acid. Now, if it would have been a protein, the wavelength would be 2850, 2900 angstroms, if the maximum would have been there. But the curve was very striking, with a tremendous maximum at 2650, and then a little maximum at a very little one at 2800, which means there's a little protein too. We had many discussions in the Genetics Society. We had summer workshops at Woods Hall. They were called gene conferences in the 30s. Thomas von Morgan were there, Barbara McClintock was there, all the geneticists came to this. And the discussion was, is the gene a protein or is it a nucleic acid? If you would have put it up to a vote, I would say 90–95% of the people would have said, of the geneticists would have said, it must be a protein. The nucleic acid does not have enough information. This was a general opinion. So when I did it in 38, on the term the wavelength dependence, and found a maximum at 2650, and clean cut, there's no question about it, it's in the books. And nobody really would believe it.
So even someone as eminent as Alex couldn't get the scientific community to believe him. So I just thought that's how science proceeds. I hope you enjoyed that. So right after the war and during the war comes the third mutagen. And finally, of course, it's a chemical mutagen. And this, of course, was discovered by Charlotte Auerbach at the University of Edinburgh in Scotland. And in fact, I'm pleased to let you know that one of Charlotte Auerbach's last PhD student is here with us in the room, Raden Schramm from the Czech Republic, right over there. So we have an interesting continuity between Charlotte and us today. She, of course, discovered that mustard gas was a germ-cell mutagen in Drosophila during World War II, but of course, because of the war, she couldn't publish it, so she made a report to the British government in 1942. Ironically, the first published chemical mutagen, which was done by her, was published in 1944, and guess what? It's an environmental mutagen. It was allyl isothiocyanate, which is a natural component of cruciferous vegetables. It's in broccoli, cauliflower, Brussels sprouts. You've eaten it all your life. And so that actually was the first mutagen, chemical mutagen published in the open literature. And of course, after the war in 1946, she finally published the mustard gas work. So this also led to other developments that came right after the war. Now we go back to DNA repair. So Evelyn Whitcomb in 1946, while she was at Columbia University and at Cold Spring Harbor, published a paper identifying the first mutant in E. coli that was resistant to both UV and to X-rays, our first two mutagens. And a few years later, Ruth Hill, also at Columbia University, published the first paper identifying the first mutant in E. coli that was now sensitive to both UV and X-rays. And also, finally, you see some women coming onto the scene in our science. After the war, there was a flood of papers that came out. Charlotte Auerbach was not the only one working on this issue during the war. And as soon as the war ended, other papers came out. One of those sets of papers was by Josef Rappaport in the Soviet Union. He, using Drosophila, also showed that epichlorohydrin, ethylene oxide, glycinol, and other kinds of compounds were also germ cell mutagens. And his other claim to fame is in 1966, he published a paper in which he coined the term supermutagens. It was to encompass all these potent alkylating agents. So he was a very important figure in this field. Also after the war in 1947, DeMarini at Cold Spring Harbor confirmed Auerbach's work in Drosophila with mustard gas. And at the urging of Hermann Muller, even in that 1927 paper where he didn't have any data in science, Muller suggested that there might be a connection between mutagenesis and carcinogenesis. So at Muller's insistence, DeMarini tested a known rodent carcinogen, a PAH, in Drosophila to see if it was a germ cell mutagen, and it was. So already in 1947, there was the beginnings of this connection between mutagenesis and carcinogenesis. It did not start with Bruce Ames in the 1970s. Also after the war, mutagenesis in microbes really started to emerge. So Norm Horowitz at Stanford showed also that mustard gas was mutagenic in Neurospora, and Ed Tatum in 1947 at Yale showed that derivatives of mustard gas were also mutagenic in E. coli. So within just a year or two after the war, it was very clear that you could take a single mutagen, in this case a mustard gas, and show that it was mutagenic in an insect and in at least 2 different microbes. And we began to draw the conclusion that there was some common feature in the mutagenesis in all these organisms. It wasn't unique to any one particular one. So the war ended, and there was fallout from the fallout. So all during the war, the US federal government was concerned not only about the carcinogenic effects of ionizing radiation, but particularly about the germ cell mutational effects of ionizing radiation. Why? Because Hermann Muller was talking to them, and they were very interested in this issue. So they paid a scientist named Don Charles at the University of Rochester in New York, all during the war, to find out if ionizing radiation caused germ cell mutagenesis in the mouse, because the government was loath to make public policy based on data in an insect. After breeding 400,000 mice at the University of Rochester, Dr. Charles had nothing to show for himself. He never invented an assay to detect germ cell mutagenesis in the mouse and learned nothing. So when the war ended, the federal government still didn't have an answer to this very pressing question. So they decided, this was the Atomic Energy Commission, and the War Department decided they really needed to spend big money, and that's when they established the biology division at the Oak Ridge National Lab. And who were they going to get to run it? Of course, Alex Hollaender. And who was Alex going to get to solve this most difficult problem? None other than Bill Russell. And Bill, of course, was also one of the founding members of our society. Amazingly, in just 3 years, in 1951, Bill published the results of his efforts. He invented the mouse 7-locus specific assay, and with it showed that X-rays indeed was a germ cell mutagen in the mouse. It was an amazing tour de force. And in just a few years, we come back to DNA repair, and Leanne Russell, along with Bill Russell, had data where they used fractionated doses and different doses of ionizing radiation, a different dose rates, that gave the first evidence of DNA repair for germ cell mutagenesis in the mouse. I also want to say a few words about Leanne. She was a remarkable person, a world-class scientist in her own right, and a real pioneer. She was the first woman president of our society, and she died just 2 months ago today at the age of 95 in Oak Ridge. For those of you who would be interested, there's a wonderful YouTube of her from last year at the age of 94, where she was interviewed for an hour at Oak Ridge. And I encourage you to go Google it, and you'll get to hear her voice and see her comments about all the issues we're talking about today. But she was an amazing person, and we owe a great debt of gratitude to her. So I want to spend just a minute about Oak Ridge. There is a fairly recent aerial photograph of this place, which some of us in this room have done time at, and nestled neatly in the hills of eastern Tennessee. There are lots of people at Oak Ridge that we should acknowledge, but I'm only going to mention 4. One of the key ones, beyond Alex, of course, was Fred Desser. In 1958, Fred invented the adenine 3 mutation assay in Neurospora. He mimicked it purposefully after Bill Russell's mouse-specific locus assay. And the reason he did was Bill's assay allowed the recovery of both gene and chromosomal mutation in the mouse. Amazingly, Fred was able to develop a similar system in a microbe. So this was the first microbial system that permitted the recovery of both gene and chromosomal mutation. It was an amazing development. And over the years, Heinrich Malling worked with Fred to improve that system. Ernie Chu, of course, invented the first gene mutation assay in mammalian cells, and again, without Heinrich Malling, it would not have happened. Heinrich was key to that development with Ernie. Abe C, of course, invented the CHO-HPRT assay in 1975, which for decades, along with the mouse lymphoma assay, is the standard assay for gene mutation in mammalian cells. And then we come to the resident genius of our society, Heinrich Malling. Heinrich, of course, invented the first tester set. It was in Neurospora. It was a series of 8 strains. He whittled it down to 2 eventually. and they reverted by certain mechanisms, by frame shifts or various base substitutions. And from that, you could infer what class of mutation your mutagen was causing. This was invented more than 10 years before DNA sequencing, and of course it served as the prototype for Bruce Ames to develop his tester set. So it did not start with Bruce, it started with Heinrich. Heinrich, of course, also in 1971, invented metabolic enzymatic activation. It was not S9, it was what I would call S30. It was a 30G centrifugation of the supernatant route over S9 plus cofactors. And he coupled it with one of Bruce Ames' strains in Salmonella to make that first demonstration. He later did it with Neurospora. So again, that invention completely changed our field, changed the course of genetic toxicology and certain aspects of cancer research. And of course, in 1989, he, along with Jim Burkhardt, invented one of the first transgenic mouse assays. And in 89, the other big transgenic mouse assay, of course, was invented in the Netherlands by Jan Goossen and his colleagues. So we owe all of these people a huge debt of gratitude. And Fred and Heinrich also were incredible organizational people to get our field, our society off the ground. So now we're in the 1950s, and more chemical mutagens were starting to be discovered, this time, of course, in microbes. So Demeritz at Cold Spring Harbor showed that formaldehyde was mutagenic in E. coli. Formaldehyde is still present in the environment. It's a common industrial chemical, so this was a big deal. And in 1958, essentially by himself, Zabolski at Rutgers did the first screening assay for mutagens. It was over 400 chemicals. He looked at it as strep resistance assay of E. coli. You can see he only found a few that were mutagenic. He didn't have S9. But this was, the reason he did this was the growing concern that there might be a lot of chemical mutagens in the environment and we better start looking. So this was one of the very first screening assays. This did not start with Bruce Ames in the 1970s. And of course, Ernst Fries, also the 50s and 60s at NIH, showed that a wide variety of base analogs and alkylating agents, other chemicals, were mutagenic in microbes. So within just a few years, lots of compounds were starting to be shown to be mutagens. Now we go back to DNA repair again and back to UV light. And in the early 60s, of course, Pettijohn and Hannawald at Stanford and Setlow and Carrier at Oak Ridge finally discovered a mechanism underlying DNA repair, in this case nucleotide excision repair, using UV. And of course, Phil Hannawald is with us here today, again showing the remarkable continuity in our field. This was an amazing discovery, and that discovery was extended a few years later by James Cleaver in San Francisco, when he showed that using UV light, that when cells were deficient in DNA repair, it could lead to a very serious disease. Obviously, this was XP. This was the first clear linkage of DNA repair deficiency with human disease. And this, of course, caught the attention of the funding agencies. So now NIH and others said, oh, maybe we should support this. And of course, this changed the course of our field as well. All of this created, among the leading intellectuals in our field, great concern about environmental germ cell mutagens. After World War II, thousands of chemicals were being introduced into the environment, and of course they weren't tested in anything. There was no EPA, of course. And Joshua Lederberg, who was one of our founding members, won the Nobel Prize, was so concerned that he wrote to the National Academy of Sciences in 1955 to express his concern about the introduction of possible germ cell mutagens into the environment, and the National Academy ought to look into it. They did not, by the way. Messelson at Harvard urged James Crow at Wisconsin to organize an NIH conference in 1966, which was the first conference on environmental mutagens. And it was held, sponsored by NIH, and brought together all the leaders in the field, specifically to discuss the issue of germ cell mutagens in the environment. Marvin Legator and Sam Epstein in '69 were the first to make sure that in a US federal document, it's called the Barak Report, named after the chair of the committee, that they proposed that pesticides be tested for mutagenicity. This is the first time that proposal for testing compounds for mutagenicity appears in a federal document. And it was very important, and this was all engineered by founders of our society, in this case, Legator and Epstein. Now, all of this science did not happen in a vacuum. In fact, it happened against a backdrop of enormous cultural and societal tumult all throughout the 1960s, and some of us are old enough to let you know that that indeed was the case because we were part of it. And so the environmental movement was one of those movements that started in the 1960s. There were oil spills off the coast. That actually is a picture of a US city that all of you would know. There was no EPA. We had tons of air pollution. Industrial waste and effluents were not treated. They were deposited directly into rivers, lakes, and streams. No EPA. Pesticides were used with impunity and sprayed everywhere. Not tested. No EPA. The Cuyahoga River was on fire and people were out on the streets protesting. So there was a huge amount of social movement about environmental pollution going on at the same time that the science was emerging. I just want to reiterate some of the key events that happened during that decade. So thalidomide was finally removed from the US drug market in 1961. Of course, this was a terrible teratogen, but more importantly, it raised the issue to the American public that, you know what? Maybe our drug supply isn't safe. Maybe our drugs aren't being tested adequately. And indeed, of course, they weren't. They weren't being tested for developmental effects or mutagenicity or anything. So this raised some concern. Of course, there was weather inversion in New York City. It killed 80 people in 1964, terrible air pollution. There were Senate hearings to remove lead from the gas in 1966. Oil spills, I already mentioned the Murac report, where our founding fathers managed to get into a federal government document that pesticides should be tested for mutagenicity. And of course, at the end of the decade, EMS was founded, and a few months later, men landed on the moon, so it was a tumultuous era. And it wasn't just this. Leading public intellectuals in this country were also raising concern about the environment. So one of those, of course, was Aldo Leopold at the University of Wisconsin. And right after the war, he published his very famous book, A Sand County Almanac. He actually died the year before the book was published, but it did not diminish the impact of his book. And in that book, of course, he argued for a land ethic, that we have responsibility, stewardship to the air, the water, the soil, etc., and that we should take care of this to preserve it for future generations. That may all sound very obvious to us today. It was not so obvious after the war. Of course, Rachel Carson, who was in fact was a federal government scientist. She was a biologist with the U.S. Fish and Wildlife Service, not far from here in Maryland. In 1962, she published her very famous book, Silent Spring. And again, she died just a year after she published that book, but again, it did not diminish the impact that that book had. It reached far and wide, not only in public conversation, public circles, but within the scientific realm as well. And she even has a section in that book on germ-cell mutagens and testing pesticides for mutagenicity. Wonder why she has those. She was talking to some of the people I just mentioned. Of course, Paul Ehrlich, who is still alive, in 1968 published his famous paper in Science called The Population Bomb, where he raised the issue of how many people can the Earth reasonably be sustained. And of course, in that same year, Garrett Hardin at UC Santa Barbara, also in Science, published his famous essay called The Tragedy of the Commons, where he raised the issue that we cannot operate on our own self-interest. It may be good in the short term for us, but in the long term, it will not be good for anybody. And that we need to work for the common good, and that the environment is something that affects all of us, and we need to operate in that way. These were leading public intellectuals. They were on the Johnny Carson Show, the Tonight Show, the Dick Cavett Show, in the New York Times. They were in the zeitgeist of this country. And in the popular culture, so this was not reserved just for the scientific community. So against this backdrop, our society was formed. And yes, that is a Saturn V rocket with Apollo attached to the top, heading off to the moon. And let me give you a few details about our takeoff. So in 1968, James Crow actually published a summary of that 1966 NIH conference on mutagenesis. And in that report, the report made 4 recommendations, and Hollaender adopted 3 of them as the founding basis for our society. And I'll get to those in just a second. In January of '69, Hollaender, DeSayers, Mulling, Legator, Epstein, and Fries met in the corporate offices of Union Carbide in New York City and basically shook hands and agreed to form our society and named it EMS. And without the benefit of email or cell phones, just 3 months later, they held the first meeting right here in Washington, D.C., just a few blocks from where we are. There were 268 attendees. There were already over 450 members of the society. And the topics at that meeting were very similar to the topics at this meeting 50 years later. There were symposia on mutagenicity testing, on mutational mechanisms, and on population monitoring. And of course, there were symposia on the mutagens du jour. At that time, it was nitrosyl compounds and cyclamates. And I would note that registration was only $10 and only $2 for the wife, and that's what it says in the program. It does not say spouse or companion, because after all, scientists were assumed to be men. I will note that there actually was one invited woman scientist on the program of our first meeting. It was Jackie Verrett here from the USDA. So we have FDA. So we have come some ways, I would think. So let me recount a few of the early successes of our society. EMIC was formed, and this was one of the recommendations of the NIH committee, and this was the first electronically accessible curated database, in this case of the entire world's literature in mutagenesis. I can remember seeing this at Oak Ridge by Liz Von Holly and John Wassam. It was all done on IBM punch cards with an IBM mainframe computer. It was an amazing tour de force, and in the late 80s, this entire database was given over to not GeneTox, but ToxLine, and this was all 25 years before PubMed. PubMed did not come on until 1996. This was amazing, and for over 20 years, our colleagues, and certainly myself, had access to the entire world's literature in a curated electronic database. That is what our society was able to do back then. In 1975, Jan Drake, who just retired a few years ago from NIHS, chaired Committee 17 report. I encourage you to go read it. It's in Science, and it laid out the strategy for the formation of this society and the agenda of what needs to be done. And I'm sorry to tell you that there's a lot of unfinished business in that agenda. There's a lot more we still need to do, so I encourage you to go read it. And I also want to note Mike Waters, who's here in the audience with us today, and he used EMIC as the literature source to bring 150 scientists from around the world together to read that entire literature, write 34 review articles of that literature, and based on that, implemented the strategy to begin TSCA here in the United States, which at EPA, which was the beginning of testing of chemicals in this country. So we owe a lot to the founding members of our society, and some of them are sitting right here in the room with us still today, 50 years later. There were other successes. Fred Astaire started the EMS newsletter, which still goes to this day. And of course, Fred and Alex started editing this famous book series called Chemical Mutagens, Principles and Methods for Detection. I'm sure all of you have seen it. Many of you have read many of the chapters in that book. And Bruce Ames used it as the occasion, as the venue, to bring his assay to the public. He published the first paper to introduce his system in 1971 in that book series. Also, of course, the Society established its own journal, Environmental Mutagenesis, with Seymour Abrahamson as the editor. And as many of you know, Alex Hollaender, all while he was at Oak Ridge, had a training program between Latin America and Oak Ridge. But when he died in 1986, that training program died with him. And one of our wonderful members, who's also still here in the audience today, Dr. Patricia Ostrowski from Mexico City, took it upon herself in 1993 to reinvigorate that training program and started what we now call the Hollaender courses, which have been going strong ever since. I want you to know that besides our annual meeting, the Hollaender courses are the most important thing we do. It is the public face of this society, and it is the educational outreach around the world that we do, and I want to thank you, Patricia, for starting this all over again for us. In the end, our founders have left us a legacy of action without activism. Now, of course it's true, Legator and Epstein, all these guys, they were working behind the scenes with all the congressmen and senators in this country to make sure the legislation happened to require testing by FDA, EPA, and everybody else. So there was a lot of action going on in that way, but they were not in the streets. And because EMS was established firmly in science without that kind of public activism, It gave the society unbiased credibility, which it has today as an advisory body. This is very important. It was also one of the first multidisciplinary societies. And all of its initial goals were accomplished, except for one glaring one, the identification of human germ-cell mutagens. And you're going to hear a little more about that from Carol Yock later this morning. So our mission is not yet complete. And the following talks you're going to hear about this morning will describe the many accomplishments of this society and what lies ahead. So there's more to come. And we owe a huge debt of gratitude to these and many, many, many other scientists for the work and efforts they did to found our society. These people were amazing scientists, and some of them were amazing politicians with remarkable organizational skills. And if it weren't for them, you would not be sitting here today, let me tell you. I leave you with this iconic photograph of our fragile blue marble home, the picture of Earthrise taken by astronaut Bill Andrews from Apollo 8 in December of 1968, just 2 weeks before our society was founded in New York City. I think it's emblematic of the legacy that our founders have bequeathed to us, that we have an obligation to protect our environment and our genome, our two most important assets. And I think that idea should animate what we do going forward for the next 50 years. Thank you very much.